Hello, everyone. Welcome to our sessions where we are going to discuss the real world impact and regulatory decision making, where we are going to gather different scientists who are in charge of getting us to know how the modeling and simulation can really impact the drug development and other strategies for registration and getting the product in the market. So my name is Gustavo Mendes. I'm a fellow in the regulatory affairs in Simulations Plus. And we are going to start our first session. And for that, I'm going to invite Frederico Martins, which is a principal scientist and scientific lead in South America at Simulations Plus. And this first session, he's going to bring some aspects on generics for emerging markets and beyond. So we're going to have 30 minutes. So Fred, please, you can start.
Good morning, good afternoon, and good evening, depends where you are in the world. So it's my pleasure to be here today with you guys and bring a little bit about our work and how our modeling simulation can support the generic market, generics on emerging market. Please, Leonardo, just confirm that you can see my slides. Then we start. So when you talk about generic, it's very important formulations in everywhere in the world, in rich countries, in poor countries, and emergent countries. But it's become very important for the emergent countries, and because the price, the cost, and everything. And when you think about which countries are the most important, we need to have close attention for the BRICS and MIST, that they are the blocks that are leading the economy for generic. On those blocks, in the past 5 years, the generic market there double in 5 years, and now it's reached almost 20% of the world share market in generics. And why this kind of formulation of this kind of medicine is very important. The first thing that you need to bring up is because the cost. Of course, the generics need to be cheaper than the branded formulation. It's become very important because sometimes the population don't have enough resource to buy the reference formulation. So the generics make more competitive the market and cheaper for them. The second thing is also very important is because when we have the generic, it's a boost for the local economy. First, because we can create more jobs, we can create more the chain, increase the chain of the jobs. Second, in the last one, it's because we need high-skilled people to work on those industries. So we need more universities, we need more training, and etc. So, however, the generic market, it's a very competitive industry because, as I already mentioned, we can have different brands, you can have different formulations for the same API. So if the industry want to be competitive and target the formulation on that market, few aspects need to be taken care. Not only those I'm sharing here, but there are many, but we can point out few of them. The first one is be aligned with the regulatory affairs, because if you're not approved, you cannot commercialize those. The second is have a selling force to make your product more visible in the market. Also, we need, as I mentioned, we need to establish a sustained local capabilities with strong local retain the talents, because to have a good talent nowadays is quite hard and it's very expensive to have a good professional. Then when you have those people in your team, you need to keep them, right? And training them even more to always increase the quality of your product. Also, we need a good product portfolio. We need to choose the right API and the right formulation for the specific market. And then in this chain, it's where Simulation Plus became to help and become very strong. The first point where we can help on the generic market and in this very complex chain. It's and we are very proud of that, is that Simulation Plus try to always training new talents, always try to improve the resource on the for the local market. And for that we have the Simulation Plus University program, where we offer for universities free access to our softwares, where the people can be training, and we can help the next generation of modders that can fill the gaps on the industry. Second, always when you commercialize our products, we offer kind of training for who has the software. So it's not about has the program, has the software in-house, but how to use. And also we offer this kind of service training the professional. And last but not least, it's the main topic I will bring here today, is our new launch that we called Plug and Play Compounds for PBBM, that we spend more time today to show you how we can use these to accelerate the generic program and make the process of production much cheaper and more competitive. So what is the Plug and Play? Is we in-house develop a list of compounds that is most likely for the generic companies, where we divide in 3 tiers, tier 1, 2, 3. For now, we have 12 compounds already qualified and validated in GastroPlus for PBBM, where all the information about absorption distribution for those compounds here you can see it's already qualified in the software. So who by this, who has these compounds, just need to use this to create the new formulations, for example. And why is very important we have pre-qualified compounds when we start the PBBM program. First, here I'm presenting to you a generic workflow for PBBM. Is not a standard or is not the unique way to work with PBBM, but here it's just a generic way to see. So always we need to think what's the aim of your model. Why do you want to do that model for pediatric, for DDI, or for only for PBBM? For that, we need to collect a lot of data, for example, PK, metabolism, distribution, solubility of your compound. In this part of the process, we spend a lot of time and money, because we need to pay for the papers. We need to spend time to collect the data and see which data is useful or not. So once you have everything in place regarding the data, we can start the data integration. That is, go to the software, the Gastro. So then there we have 3 basic blocks, where you have the drug parameter, have the system parameter, for example, is the physiology. If you work with, for example, for pediatric population, you use the physiology for this population, or if a disease population, etc. Also need to the clinical design of your data, PK data you are finding and are used to develop your model. So once you have all these 3 blocks here in place, you move for the model development. It's a very important process because it takes time. Sometimes the model doesn't work in the first place, so you need to refine the model. You need to find more data and find solutions to make the model work with the reality. Then once you have the proper PBPK model, so we say they have a qualified model, then it's important because we know that the model is not always 100% perfect. So we need to show where the strength of the model, where you can improve this model. Now, once we have this, we can say that we have our final PBPK model to move for the PBBM strategy. So once this model is in place, the PBPK, we can take 2 directions and for PBBM. The first one, we can use the Gastro for exploratory analysis to see, for example, in the early stage of formulation development, you can use to see if your new formulation, if your new release is appropriate or has a good chance to be bioequivalent or not. However, if you have a more clinical data, you have more in vitro, in vivo data, we can move for another aspect for more regulatory application, where you can try to do IV-IVC or IV-IVR and have your PBPK model. In PBPK model, Simulation Plus can bring to you two different strategies. First, we can use the DD Plus and combine with Gastro Plus, for example, to specification of the solution method. Sometimes when you are developing a new formulation, you don't have idea, for example, which median, which sprint speed you need to use. So this software, this package can help you to start to develop the solution method. Also, we can have the safe space that you know how much the solution, how much your formulation can differ from the original one and still be bioequivalent. And as I mentioned, also you can design the formulation, right? You can include more recipients or reduce, change recipients and see how this will impact on the solution, how this will impact in the bioequivalence of your product. So how the plug-and-play works, as I just showed you, we need a lot of time to qualify the model, to have a good model for PBBM. And after after we finalize those models, the user of the plug-and-play only need to, not only need, I mean, but what you need to do is have a biopredictive solution method for your drug formulation solution. If you have the DD plus, you can explore this more. For example, as I mentioned, a safe space. Then you have the in vitro solution in biorelevant medias, for example, where you can predict well the PK. You plug this in gastro, and you can have bioequivalence. Then you can have different trials in different population with different variabilities of intra-subject variability, for example, and see if you have a good chance to approve this new formulation as bioequivalent as generic. And what are the benefits using plug and play? The first thing I would like to mention, it's because is transform your R&D program in higher performance program, because you can skip steps. You can skip the time you spend for the qualification and development of your model. So you don't have to use your research to burn your brain to develop the model. You already have this in place. The second I would like to mention is you can reduce the cost of data creation, because as I mentioned, when you develop the model, you need to collect the data, and all those papers come from paid publication, and it can cost a couple of $1,000s to have only the data in place. And this step you don't need anymore because the model is already qualified. Also can reduce the cost and time with training, because you don't need to go through all the process of training. The model is already qualified, so it's making much easier for the user plug and play and use what is there. Increase the efficiency of the R&D program, as I mentioned, because all those points. And one of the most important aspect of this plug and play is all our models are qualified and the reports are pre-reviewed by experienced regulatory professionals. So it's to increase your chance to approve your submission with regulatory agencies. So it's quite important. And now I will just show an example with one formulation, one compound we have for PBBM, that is Zolpidem ODT formulation, and see how we can use the plug and play to accelerate the R&D program. So Zolpidem, you can have different type of salts. So this one we use mutated, and it's a compound with molecular weight about 800 grams per mole, but the free base is about 307 grams per mole. The salt base ratio is 1.24. We have a good permeability for this compound in CaCO2, that's 4 power minus 4 centimeters per second. Log P 2.4, it's a weak base, pKa. And here I bring to you one of the screenshot of the gastro where we care about the quality of our data and how we use this data published or data originating house for the industry in our models. For example, here you can see the pH, the solubility versus pH, and then you use this curve to set up our predicted pKa for our compound. Also, we care about all the information that is inside the model. For example, the physical chemistry property. Always we double check. We have QC of our data for different professionals to make sure that what is delivered there, it's in high quality and is aligned with the reference data. So when you have enough data, for example, in this compound, in this model we have, for example, we have the solubility of the salt in different medians, biorelevant medias. We have the precipitation time. So then you can use the dissolution models to see how the dissolution happened for this salt. Also, we care about how the clearance process is taking place in the model, because we need to use sometimes full PBPK or use compartmental model, but the enzyme kinetics for those is important, because in the GI tract we have different expression for different enzymes. So if the release happen in one part of the gut, maybe the bioavailability will be different or not. So we check that and qualify this information, this data with against the observed PK. Also, we try to first qualify our models with IV data, then after that, when it's possible, of course, and after we move for oral data with different formulations in those, and always compare when it's possible different formulation, for example, method release, solution, syrup, or supersaturation, for example, in different dose to check if our model can predict the linear range or not. As already mentioned, none of the models always 100% perfect. So we need to show what is the weakness of the model and how we can improve that, and also using sensitivity analysis to see which physiological parameters can has impact in the bioequivalence of your drug. For example, for the zolpidem, on one of the most important parameters we found in sensitivity analysis was the percentage of small intestine and stomach transit time, stomach pH, because the absorption and the solubility of these drugs depend of the local pH. Then you can see in this sensitivity analysis that our model can predict that. Then now once the model is ready, here's where become from here, let's say the user need to care from here. That is how to input the solution profile in the software, etc. And this example here is one of the classical example. For example, when we have two formulations, but they fail for F2 comparison at pH, specific pH. How you can see here, the reference and the test formulation, we have different release at pH 6.8. However, the question that remains is this difference that makes the fail, the F2, will be sensitive for the absorption, will be sensitive for this difference. So the PBBM will help us to identify if it's sensitive or not, if it's become important or not. Also, in the absorption part or the dissolution part of the software, we try to be always mechanistic process and integration of the dissolution. For example, we have the mechanistic solution. We can plug the particle distribution size to see how it's behaving, etc. One of the most important part of when you tackle in virtual bioequivalence, it's what's the variability, because the variability determine the size of your population. For this example in a specific, we know that So P has about 30% of within subject variability of 30%. Anyway, when you check how big should be our population for clinical design with this variability, we find out that should be 32 subject for a crossover design 2x2. So also the Gastro has the flexibility, and we can check all variables that's plugged in the software. However, if needed, the user can, to mimic a specific population, you can change this variability as needed. But many of those parameters is already qualified, and you know that's work well. So also you can create different trials with different sample size and etc. If you want, you can simulate physiology intersubject variability, or you can apply standard value for that. So then here it's how it's work. So once we plug the solution on the software and you cross all processes I mentioned, then as a result, you have the PK curves and the statistical analysis for many trials. In this case, I run 10 trials for 2 formulation. That what we come up with the conclusion about if the F2 was decisive information for the bioequivalence. That is not that the absorption for this formulation was not sensitive for those types of the dissolution. So then you have a higher likelihood to have a positive bioequivalence for those 2 formulations. Then the message that I would like that you or us take home here, that these plug and play and GastroPlus are powerful tools when combined, that the mechanism dissolution, the mechanism absorption, you increase the potential of the benefit of PBBM and PBPK. And for sure, the plug and play will enhance the R&D program of your industry. So thank you very much, and let me know if I can help with something.
Thank you very much, Fred. Thank you for sharing this very important initiative that Simulation Plus is bringing, and also to share a very interesting example where we have a success case on predicting and using the PBBM and the virtual bioequivalence in generic development. So we have some minutes, and I'm gonna give you some questions I received from the Q&A. I just want to remember all the attendees that we're gonna have a panel session later today after all the presentations we're having. But since we have some time for questions, I'm gonna give you, Fred, some that came here. So the first one that I want to bring you is regarding validation. So Fernanda asked about the validation on these plug and play models. So how is the validation step? The observed data from different clinical studies are provided. How do you consider the model to be validated for those drugs that are already available in the plug and play?
Excellent question, Gustavo and Fernanda. So always when possible, we prefer to start our model and our qualification development using IV data, right? But it's not always is possible. It's not always all drugs have IV data already published. So then we use most simple formulation, for example, solution formulation. Then we qualify the model, use every single data we can find published. We tackle in the database of Google or Washington database to see what is out there for this compound or for many different types of formulation to qualify our model. And how we say that the model is qualified or not, we always compare the visually how it's behaved, the curve of our simulation with what is observed, if they have the same shape of the same inclination, for example, the same or similar, at least similar final phase, etc. And also compare the numeric of the data, for example, AUC, Cmax, volume of distribution, terminal clearance, and etc.
Thank you. So this is very important because many of the people that intend to use this kind of tools are worried about how wide and how different sources are included in order to consider what they're going to have in their own facility with their own formulation, because we know that generic in many cases in Brazil, for example, we know that excipients may be different from reference and manufacturing and equipment. So that's why validation is something that is really important to understand and to think. I remember one question also that I want to give to you, since we have some time, Fred. That is regarding so we have a limited number of drugs, right? So is there any plan to expand this? How is this work going on?
Yes, we are. Thanks for the question, Gustavo. We are working hard always to expand this list of drugs, and we are in touch with our clients if the market what they need. For then in the future, we can expand this list. But for now, what we have qualified in house, ready to go, is this exact list.
This is exact list, yeah.
But
please go ahead.
But of course, if you or any of the attendees here has a suggestion for compounds, please send to us by email. We'll be more than happy to have a look and see how we can support you in the future with plug and play.
Yeah. So one last question is from Mohammed that is bringing actually a request regarding and wanting to know the virtual bioavailability related to submission success in generics. So so by the case example, we have seen one case example of success, right? So but this is maybe a regulatory question also regarding how do you see regulatory agencies accepting and discussing this as a possibility to be included in the submission of dossiers?
Sorry, Gustavo, I think I didn't get your question. Could you?
So how is the acceptability of virtual bioavailability or virtual bioequivalence by regulatory agencies, and how they are discussing it?
Okay. Yes, it's a good question, and thanks. It's the agency around the world, EMA, FDA, and Visa. They are accepting this kind of work. Of course, it will depend the quality of the model, how the model was qualified, the documentation you can provide. It's not only about you have the final simulation, it looks nice and you have a chance. But we have many success cases where the virtual bioequivalence was used for regulatory application. But where I can tell you too that we don't need to think always only about application for regulatory aspect. For in-house, for development of your formulation, it's really, really important. So sometimes you don't use the model for regulatory application, but you save time in your R&D. So where it's become very strong PBBM and virtual bioequivalence, you can avoid mistakes in your clinical study, for example.
Well, that's great. Thank you, Fred. Thank you, everyone, for attending. So we're looking forward to meet you all again in a few moments for the last session. And also, I invite you to come for the next session, which is going to be with Xavier, and we're going to discuss more case examples and applications of these tools in the drug development. So thank you very much. Thank you, Fred. See you soon.